Well, I got nothing, so go ahead.
Alrighty, well, we're going to kind of go over the quizzes, and not everyone has finished all the quizzes, of course. That tends to happen that way sometimes, right? So pretty much what we're going to do is I'll try to go over everything in a way that doesn't just completely give away the answers. And I said I'd give you guys through Wednesday, so I'll let you finish quizzes by the end of today. But after that, then it's too late. We'll put the test up today. I've almost got it finished, but not quite. So just expect to see an announcement later on this afternoon about the test and when it's up and available for everyone. And I've gone through all the quizzes. There were a few mistakes that I made, and I'll try to highlight those. Of course, if I make mistakes, I'm not going to hold those against you guys. So, all right, let me get this together here, and we'll go ahead and go through a little bit of review. I just want to make sure that you guys know what issues were with quizzes and know exactly what's going to be on the test. And there were a couple of mistakes made on some of those quizzes, and I'll give you guys credit for those. So we're going to talk about first, kind of going back to quiz 6, chapter 6. We covered a lot in this unit here, so apologize for everything that we've put into this one unit. Really should have split that up. So got everything in order here. All right, you guys see my screen there?
Yeah.
All righty. Okay, so first, a couple of things. As you guys probably have realized by now, I like to get a little tricky on some of the questions here. So one of the early questions there was about what classes, categorizing plants, what they fall into. And notice on the PowerPoint here that it says major food, fiber, wood, and ornamental plants, belong to two main classes, the gymnosperms and the angiosperms. So the question was something about, I said all crops belong to these two classes. And if you remember in the presentation we talked about it, I was careful to try to point out that although the vast majority of our crops do fall into these classes, not every single one of them does. Of course, it really depends on how you view a crop. And I think it's important for you all to note in your professional lives as agriculturists, whatever branch of agriculture you end up going in, that there are so many different categories of crops. A crop is really anything that you can make money off of, right? I mean, anything that you can figure out a way to provide a service or sell a product from could be a crop. So not every single crop falls into these two main classes, the gymnosperms and angiosperms, which again are kind of like the coniferous plants and the flowering plants. We could have mosses, for instance, or ferns that are ornamental plants that could be used for horticultural purposes. Technically, those could be crops. So just remember that and apply it. I think one of the most important things for us in agriculture is to find what your potential niche is. How can you, do something different than everyone else, really? How can you figure out how to make money in the world of agriculture? And it requires you to be creative in many cases. So don't limit yourself to this, couple of sentences here. how most of our major food, fiber, wood, and ornamental crops belong to these two classes. Get creative. Next thing, there are some mistakes on the matching part of monocots and dicots. So we shorten that, of course, monocotyledons. We're referring to that first seed leaf that pops up as the seed germinates. And there are two major differences there. One is in the venation of the leaves. Also, the floral parts are slightly different. Just know how to distinguish monocots and dicots. Of course, we have crops in both categories. All of your grasses are monocots. Almost all of your more ornamental flowering plants are dicots. So first it's the cotyledon, but there's more to it than that. The venation on the leaves, the number of flower parts as well. And this is a actual class. So if we're going into the actual taxonomy of plants, you go kingdom, phylum, class. So the phylum here is the angiosperms, the Anthophyta or Magnoliophyta, and then the class is monocot or dicot, basically. So really important to be able to distinguish between plants, and as we talked about in later presentations, the different photosynthetic pathways and different needs for monocots and dicots. They often have different production systems too. So, and for your crop project, be sure to distinguish whether it's a monocot or a dicot. Next one here, this was a mistake that I made. There was actually a question that repeated on there. Not exactly sure how that happened, but I think I was intending to change one of the words there. I just wanted to be really clear on this. Of course, remember the different meristematic regions. There's like that apical meristem at the top of the plant, kind of the shoot meristem, where most of the hormones are concentrated. That's like the growing tip on the top of the plant that creates the apical dominance and makes dominant leaders in plants. But there's also meristems in the stem itself, as well as in the roots. The stem the meristem in the stem is actually in the cambium itself, the vascular cambium, which produces the new xylem and phloem, but then the cork cambium, which chiefly produces bark. And this is mostly in woody plants. It's actually really important to be able to denote and tell where the vascular cambium is for propagation, especially for grafting. So if any of you are interested in like orchard production, or if you're doing, I think someone is doing something on apples, when you talk about grafting, one of the biggest issues with grafting is matching up the vascular cambium of your scion and rootstock. So anyway, vascular cambium produces xylem and phloem. The cork cambium, which is the secondary cambium up here, produces mostly bark tissue. So, back to the monocots and dicots here. This is just the order in which the slides are in. Strap-shaped leaves and parallel venation on monocots, whereas dicots have broad leaves and net venation, where the veins are radiating from one central vein. So remember that. Another tricky question here. Of course, all plants do have some optimum temperature, right? That's not just for the growth, but also for the germination of seeds. So for the actual maximum vegetative growth and then flowering of plants, they suggest here that most temperate region plants grow between 39 degrees Fahrenheit and 50 degrees Fahrenheit. And those are the generally the limits of temperate region plant growth. I think that's a little bit misleading, and I tried to highlight that there, that is kind of an optimum temperature range, but it doesn't really specify the optimum temperatures for particular crops. So on the question, I put all plants have optimum temperatures, true, for growth and flowering, and that they are all between 39 degrees and 50 degrees, and that is not true. So just note that. I know that was a kind of tricky question, a little bit misleading, so I wanted to clarify that that's where we get the most growth in the temperate region plants. But really, you need to look at optimal temperatures, again, in the multiple categories. One, what is the optimum temperature for the germination, which, again, there's going to be a minimum, an optimum, and a maximum for germination, as well as growth for plants. So knowing those for your particular crops is extremely important, because it says what is a cool season and what is a warm season crop. So again, kind of a misleading question, but just understand that each crop has its own optimum temperature, and they're not all in this 39 to 50 degree range. We've grown tomatoes in hoop houses and monitored temperature at different levels in the hoop house. We found that at the very top, our hoop house, of course, was not, we didn't have any electricity run to it. It's just for season extension. So we were not actually ventilating at the top. We had side vents that rolled up and down. So that hot air was actually getting trapped at the very top, and our tomatoes grew in kind of, even though it was outside of this optimum temperature, they were still able to produce, even when that temperature range was well over 100 degrees. We had days when it got up to 120 degrees, and still plant growth was not limited at that point. So I just think that is a little bit misleading, that is the greatest range for growth for temperate region plants, not for tropical plants, right? So just note that. Many tropical plants, for instance, they really slow down and might not even grow, under like 45 degrees. So it's just a misleading range. I want you guys to recognize that it's different for each crop, and you can't really put all plants in one category. A little bit on the difference again in determinate or indeterminate. I think there were a few that was a matching question, and a few of those most people got wrong. So one, indeterminate growth, think of it like, if we're talking about tomatoes, for instance, as like a bush type versus kind of a vine type tomato. For determinate type, it's basically that terminal shoot is going to grow until it's able to reach maturity for the fruiting crops, the tomatoes themselves, and then it's going to stop. It's going to ripen the fruit, and then the plant's going to senesce. So for the determinate plants, there's really kind of a short and very defined growing season for those crops, whereas in the indeterminate plants, they continue to grow throughout the season until they are stopped, often by environmental influence. Every once in a while, there might be senescence caused by other things, just like a maximum fruit set, for instance, or a maximum yield. There is particular life cycles for some plants too. So indeterminate doesn't necessarily mean that it's just going to grow forever, but it's going to continue to grow throughout that range of temperatures that it can produce on. So, and they looked in that slide in the book at tomatoes on the left as having an indeterminate growth pattern, but I just want to point out that not all tomatoes have that, right? We have both bush and vine types. Beans are the same way. We actually have kind of three different types for beans. We've got pole beans, which are indeterminate. We got bush beans, which are determinate. And then there are also like the half runners, which kind of do a little bit of both in a way. It's also important to note that for harvesting of crops, indeterminate crops, while they produce for a longer season, and you can get usually a higher yield throughout the season with them, you really cannot mechanically harvest them. You have to hand pick indeterminate crops. So in a lot of commercial agricultural settings, most of them are determinate crops that can be mechanically harvested. At one point, we visited one of the largest tomato growers in the country in Central Valley of California. And they had a really interesting production system where they basically combined all of this different machinery into one implement. So they were able to plow, disc, and till, and plant, and fertilize, and put irrigation in all at the same time with one pass. Also, actually, I'm sorry, they also laid down row cover too. A mulch, plastic mulch down too. So it's like doing all of this in one pass. It was a really highly mechanized farm. It was like 10,000 acres, but they only had actual 10 agronomists. So like one person is over 1,000 acres of production. Pretty interesting. But they could only grow the bush type, the determinate tomatoes, because they were also doing the same thing when they're harvesting. They had one machine that harvested it, pulled up the plant itself, and also prepared the soil for the next round. So it was a fascinating system. But again, bush type determinate crops used mostly for mechanical harvesting because you don't get a maximum yield all at once in an indeterminate crop. Just want to highlight that there. A lot of people actually miss this one here on the pollination front. Fruit, grain, and seed formation begins with pollination. And some people put like fertilization down there, and I think I'm going to actually go back and count that because they're pretty close there. But it's all initiated by pollination. The actual pollen, the male sex cell landing on that fertile stigma, that sticky receptive part of the female plant, or female flower, I should say. And of course, recognize the difference in self and cross-pollination and the differences that come along with that in terms of genetics and how the genetics are passed down. Seed saving, it's important to know whether it's a self or cross-pollinating plant. Next, getting into the issue with amino acids. Another tricky question here. I said that there are 16 essential amino acids for plants. Well, no, that was a total lie there. There are actually 20 different amino acids found in proteins, but only 8 of those are actually essential to plant growth, and they are listed there. So just know that there are 8 essential amino acids for plant growth, and well, really for protein synthesis, although there can be up to 20 for really the best health of plants and people too. Of course, these essential amino acids cannot be synthesized by animals. They can only be synthesized by plants, and so an animal has to obtain it at least first, initially, from plant protein. Of course, as you know, we have carnivorous animals out there that do not eat any plant matter, but they eat an organism that ate the plant matter. So again, they are getting those essential amino acids in the form of proteins at some point from plants, whether that's direct or indirect. The part of the plant that contains the most protein, this is very important, is of course the seed. Seeds, there are really 2 parts of plants that have the highest caloric content. and that is seeds and roots. Generally, there aren't too many calories in things like leaves or shoot tissues. So like you eat some lettuce, you're not really getting much calories. You're really not getting very many proteins at all. You can get amino acids from those parts of plants though. But generally, most caloric content is either in seeds or in roots. A close third would be in fruits themselves. But in terms of protein, it's mostly in the seeds. So, and a good portion of a seed is actual protein content. So really, we actually eat a lot of seeds in our diets. All of our grains are seeds, of course. In some cases, we eat fruits that have seeds within them, and we get a lot of that energy and nutrition from the seeds themselves. And if you think about it, really, the plant, through its life cycle, is storing most of the energy in the seeds. It's the plant's goal to reproduce. So as it produces, it's going to allocate most of its energy when it gets to maturity into seed production. And if you think about energy and respiration, where that energy goes is where we're able to obtain energy. So usually in the summer, sometimes in the fall, we're harvesting crops in the form of seeds. In the winter and spring, sometimes we're harvesting them from roots as well as in the summer as well. So again, two plant tissues that have the most caloric content are seeds and roots, or modified root structures, but the most protein is stored in the seeds. It got a little bit confusing on DNA versus RNA. So know the difference between DNA and RNA. They are very similar, but DNA is kind of what are the active genetic material that can replicate itself and transmit information to other parts of the cell. But it's the RNA that is actually the template for DNA that allows it to translate. So you have to have both DNA and RNA. It's important to recognize the similarities and the differences between RNA and DNA. So the RNA actually does control the cell growth because it is how it's translating and causing different DNA to be, again, it's the translation template to be transcribed. So DNA is self-perpetuating genetic code, but it's the RNA that actually controls the cell growth. DNA being double-stranded, RNA being single-stranded, RNA having uracil instead of thymine. And I can see where you might get confused here because it says the DNA molecule acts as a template from which a complementary strand of RNA is formed. But RNA is what is actually used as the template for the DNA to reform. And I'm going to go back and alter some of the questions, or sorry, the answers here, or the credit given to some of the answers here from the difference in a gene, what a gene is, right? So the gene is the ultimate hereditary unit, and it is a sequence of triplet organic bases. So some people put, it was a fill-in-the-blank question, some people put gene, which was correct, the correct answer I was looking for. Some people just put triplet or organic bases in there, and I'm gonna count those as well because that could have been slightly confusing. But remember, it is the gene itself that is that ultimate hereditary unit. Of course, genes can be linked together. And often it is several genes interacting together that can determine the different characteristics of a plant. And when we have multiple genes on the same chromosome, they are termed alleles. So the alleles are kind of how the genes interact. And again, the alleles could be dominant or recessive, and they can be homozygous or heterozygous. So homozygous being that they would both be dominant or recessive, both alleles affecting a trait, or heterozygous being one would be dominant and one would be recessive. So that's important to know. Heterozygosity, again, is the difference of alleles. And as genetics are passed down through generations, these alleles can be altered. And there is depending on whether they're homozygous or heterozygous. So when you have an F1 hybrid, generally the F2 generation, that the next generation, if you were to save those seeds, is heterozygous, which causes it to be different than the parent plants. So that's the reason why we really cannot save seeds reliably from F1 hybrids, because the next generation is going to be different, always. Of course, there are many reasons to have hybridization still. We have hybrid vigor itself is a big reason. We have certain traits that we can only get in hybrid production. So, I mean, there's importance to F1 hybrids. It's just that we really cannot save seed from them reliably because of the differences in the next generation. I will say, though, that one thing that I found when I was doing an independent study in plant breeding when I was in college, and the person I was looking with, he's actually probably released more varieties of vegetables than any other person living. He did what he calls dehybridizing the hybrids. And in his opinion, he said hybrid varieties are just, incomplete work, basically. Because you have to really take selections, new varieties to a stable generation, which is often like an F5 when you start to get stable heterozygous or heterogeneous crops. And so what he did, and he made a lot of enemies doing this, is he took a lot of hybrid varieties and he just continued to select them until they became stable. And then he released those varieties to the public domain. So a lot of people didn't like him for that, but I thought it was a good work, something good that he was trying to do, because he was trying to make those useful traits available to a lot of different people. One of the things that he found, though, in doing that was that a lot of F1 hybrids are just termed F1 hybrids because they want you to continually buy the seed from the seed companies and not save them yourself. So he found that in some cases they were already stable. They were called an F1 hybrid, even though they might have been like an F5 generation, just to try to fool people into continually buying them. So anyway, don't always listen to that. I mean, I'm not going to say that it's not that you can't save seed. You can save seed from F1 hybrids. Just know that it's not going to reliably produce offspring that are similar to the parents. However, no harm in just trying that out every once in a while. Another thing here on mutations. Mutations are always bad, right? Well, no. A majority of mutations are negative. It can have negative effects on plants, but we can actually have really good effects from mutations as well. Some really novel characteristics appear as mutations, and whole genomes actually change as a result of mutations in many cases. So mutations are not always bad. They're kind of a result of just that naturally occurring replication of cells. There are errors in nature, right? So, and that's not always bad. So I just want to throw that out there. Mutations can be good too. When we talk about seed and what we do with seed, we have a few different methods of preparing seed for to actually overcome dormancy of the seed. Dormancy is extremely important because it allows us to actually be able to store seed. But and most of our crops, we don't really have these issues, but there are some specialty crops that you do have to actually soften that seed coat, that outer layer of the seed that is often extremely hard and impermeable in some cases. Sometimes you have to remove the seed coat in order for it to actually be able to take up water and germinate. So we use scarification as a way to do that in some cases. And that's where we are actually physically altering that seed coat or chemically altering. So heat treatment and using acid, those are both methods of scarification. We also have stratification, though, and that's important, especially for like native temperate plants. Stratification is usually a longer period of time where you're exposing it to like a cool and moist environment. Of course, you still have to have adequate oxygen, so you don't want to just like stratify seeds in water, for instance. But usually it just mimics what the plant goes through naturally in its natural habitat. So scarification, physical altering of the seed coat; stratification, should say on scarification, physical or chemical alteration of the seed coat; stratification, a cool, moist, chilling period over a period of time. A lot of our native plants have to be stratified. And you can do that often, sometimes just by keeping them like in a container of like peat moss outside. Or some people put them in a refrigerator with moist peat moss or something like that, or store them in a freezer for a period of time inside of the fruit. So different ways of stratification, but just note the differences between scarification and stratification. Most people got this correct, but I still want to point out the importance of germplasm itself. And again, some people just put protoplasm down, and I'm going to count that. But really, germplasm is the protoplasm that contains reproductive cells, and that contains the genetics. So, and sometimes that can be in the cytoplasm, which some people did put cytoplasm down. I ended up counting that too. But really, germplasm could be anything that has that protoplasm in it. It could be seeds. It could be just actual cuttings of the plant itself. If you're using like tissue culture or micropropagation, it could just be a piece of the plant. So it's anything that has that protoplasm and contains really just the DNA, the chromosomes of the plant in it. So germplasm really could be any part of the plant, technically, but often it is looked at as seeds. Moving on to water, wanted to again look at the whole issue of water potential, and that is just basically the power, the energy of the movement of water through anything really, but for our purposes, we're talking about it in terms of soil. And water naturally, again, flows from high to low water potential, moving across a wetting front. That wetting front is making it to where all the soil has to be, or at least that same level of soil has to be moist in order for it to be able to continue to progress through any type of flow through the soil. So noting that saturated soil with the atmosphere at 100% relative humidity has a water potential of 0. But is that actually available water? No, it's not. That's the gravitational water. So remember, you can't really obtain water plant roots can't get water out of saturated soil. It has to at least drain off. You have to have some oxygen in the soil for it to be able to actually take water up into the xylem in the roots. So a little bit more about that here in a minute. Did want to look again at the whole thing with fertilizers and salts. So, and I want to clarify too that I hope I didn't mislead you guys on this. Saying that fertilizers are always salts. Well, that's not true. All synthetic fertilizers are bound to salts. There are many different types of fertilizers that you could use that are not necessarily bound to salt. So I didn't want you guys to get confused and say, all right, well, fertilization is a bad thing because it's based on a salt and it can create osmotic energy, which can prevent water movement into the plant. That is true. Fertilizer can be a salt and can inhibit water uptake if it is, continually stored in the soil without flushing it out. So, and you can see that often. Sure, some of you probably have a grandmother out there who has lots of house plants, right? Somebody out there has one. Megan, you got one? If you look at those house plants, a lot of times you'll see them. My grandmother was guilty of it. She would use things like Miracle Grow constantly. She wants her house plants to look good. You can actually see the salt buildup, especially in containerized house plants. It'll appear with the Miracle Grow, you would actually see the blue salts accumulating at the top of the soil. You'd also see white salts as well. Doesn't have to just be from fertilizer either, though. It can be from anything taken up and accumulating in the top of the soil. Lime deposits, for instance, can kind of do the same thing. They can actually cause burn, as we looked at in that picture or that illustration there. Some sort of abiotic issue there due to over accumulation of fertilizers. So fertilizers are salts, right? Salting soil is a bad thing. Remember, the Romans actually used to do that when they conquered people. They would actually salt their soil so that they couldn't grow crops, and they had to rely on them. But we are inadvertently doing that when we over-fertilize. So just the use of synthetic fertilizer itself is not a bad thing. I want to throw that out there. It's the overuse of fertilizer and the accumulation of those salts in the soil that can inhibit the water uptake, and in some cases even pull the water out of the roots. All right, back to the water potential. Most of you got this correct, but there were a few people that missed this, so I wanted just to review that. So I could see where you would think that at a water potential of 0, that would be easier to overcome for plant roots to actually be able to take up water. But just recall that at a water potential of 0, the soil is saturated and you can't actually take up water. So at field capacity, it is usually around 0.33 atmospheres of pressure that you actually tend to get the best, air to water ratio in soil and the most available water. So water is not available at 0. It is available at slightly below 0. And as water continues to dry out, so here we have it right here at negative 1/3 of an atmosphere and up to negative 15 atmospheres. Anything beyond that generally is too difficult for plants to be able to take up. And again, available waters between fill capacity and the permanent wilting point in plants. All right, moving on to the last part here, talking about plant nutrition. Again, we're looking at ionic forms of plants, and some plant nutrients have different ionic forms of the different mineral elements found that can make them easier or more difficult to be taken up. I just wanted to, again, point out the importance of just recognizing what these different ions are, and note that some of them act synergistically with each other and some act antagonistically with each other. So it's really a balance of nutrients that we need. You can definitely over-fertilize, right? And again, an element is considered essential if the plant needs it to complete its life cycle. And there are 17 essential elements for the growth of most plants. There are some plants that might need some other micronutrients as well. Some plants that won't need all of these, especially when we're looking at things like nickel and zinc. Not all plants need all of them. So this is like the widest variety. I made a mistake here on that last quiz on a matching. So if you saw that, yeah, most of a plant's dry weight is carbon, hydrogen, oxygen, again, that we're not getting from mineral nutrients, but they're actually getting from CO2 and from water. So we're not really supplying these so much to the soil as plants are able to naturally get them. However, carbon, hydrogen, and oxygen can be obtained from organic matter. So do note that we can amend the soil to add carbon, hydrogen, oxygen, but that's not really fertilizing per se. It's just more amending the soil itself. So the one thing I did want to point out here, though, that the error that I made, I put that the elements listed, the primary macronutrients listed on a bag of fertilizer, they are NPK, nitrogen, phosphorus, and potassium. And on the quiz, it incorrectly put carbon, hydrogen, and oxygen on there. That was probably my fault. So I'll fix that. So just want to reiterate that, yeah, it's NPK on the fertilizer bag, right? Your 10-10-10 is 10% nitrogen, 10% phosphorus, and 10% potassium. And know what the primary, secondary macronutrients are again, and the micronutrients. I think most of you got those correct. It's important, and I think I mentioned this in the presentation initially, but a lot of people got this wrong, so I'm going to change that as well. What is actually responsible for the dark green growth in plants is nitrogen. That is because it is a main component in chlorophyll. Magnesium is also in the chlorophyll and is important, but there's a lot more nitrogen in there than there is magnesium. So that's what is mostly responsible for dark green growth. Of course, mobility is very important, and that was something that a few people got incorrect on the quiz about. One, about it just being important, but the other thing about which nutrients are mobile versus immobile and how deficiency symptoms appear as a result. So let's just go back real quick through these. Nitrogen, again, is found in all proteins and is the most commonly deficient in plant systems, to the point to where if you test soil, if you send a soil test off to Virginia Tech's soil testing laboratory, for instance, they don't even give you a nitrogen analysis because it's inherent that you have to supply nitrogen in some form. Responsible for dark green growth, we talked about nitrogen fixation, how it can occur in some plants. You can actually get some nitrogen from legumes and other cover crops that are then grown and then plowed under or terminated before they go to seed. again, because they're putting most of their energy in the seeds. And if you let them go to seed, then you're going to lose that nitrogen through the proteins in the seed. How it looks differently in synthetic versus natural nitrogen fixation. Everybody pretty much got that, but the different forms of nitrogen, ammonium, NH4 plus, is a cation. Nitrate, though, NO3 minus, is an anion, which allows it to be more easily leached from the soil. So nitrogen deficiency we often see is yellowing of the leaves, chlorosis of the leaves, again, because it is this primary component of chlorophyll. Phosphorus, really important, and also often the second most limiting element, and can easily be supplied through adding organic matter to the soil often, and then having a good living soil. It is not responsible for green growth. I just point that out, and I think there must have been something in here that was confusing, because a lot of people miss that one. There are mycorrhiza fungi that can help plants to take up phosphorus, make it available from the organic matter. In living soil often allows phosphorus to be more available. So this is probably the part that got people confused, because it says phosphorus deficiency show up first in lower leaves, which are characteristically dark green and red or purplish in color. That's from the deficiency. So nitrogen causes dark green growth naturally. A phosphorus deficiency can cause there to be dark green growth. So I'm probably going to go back and change that and count that. Most people did miss that, but it often kind of appears as red or purplish as well. So dark green we usually see as a good thing, but dark green along with red or purplish color is a phosphorus deficiency. Potassium, important for transpiration because it controls the opening and closing of stomata, and can appear as chlorosis and necrosis. It starts at the margins of the leaves in dicots, and then in monocots starting at the tip and then along the margins of the leaves. I think you guys are good on the calcium, but again, think of calcium in plants kind of how it is in people. It's for the structural components, tissues. And when we have deficiencies, we often have these tissues kind of appear to be rotting. Magnesium deficiency, intraveinal chlorosis of lower leaves. Magnesium is mobile. So that's why we see it on the lower leaves, because plants will reutilize it to move it to new leaves if they need to. Iron is the one that, or one of the nutrients that is immobile. So that's why we see that chlorosis appearing in the upper foliage, because plants cannot move it to the new tissues. Again, when you're looking at plant problems, you got to look at multiple measures. You have to look at all kinds of different things, really, because of the compounding issues of deficiencies and imbalances of nutrients and how they act together. So performing like tissue analysis or just trying to correct one nutrient at a time at a small amount on a test population is a good idea to try to really see what was deficient. And that's, again, due to at least the mobilization, the remobilization of nutrients. They're always going to be putting that, if it is a mobile nutrient anyway, into reproductive tissues. And I think that's all of the answers or all the questions that were most missed. Did anybody have any other questions or anything that they saw that in the quizzes that they were not sure about or needed more clarification on? Any other questions, comments, or concerns? You guys ready for the test?
Kinda. Maybe.
You've got it. And remember, I mean, you've got access to these reviews. It's on Blackboard. It's not going to be a lockdown browser or anything. It will just be, it will be timed, so you'll have 2 hours to take it. You can actually stop and come back later if you run into something. So if something happens, then you can hit the save button at the bottom. You can come back later and finish it. So it'll be open today, and it will be open through next Wednesday. So get on there. I'll put the announcement as soon as it's up. So good luck. We'll see you guys next Monday.
Yes. Okay. Be safe.
All right, y'all have a good one.